Hello everyone, I'm preparing a tutorial to show how I have recorded my lesson plans. I start with a PowerPoint file, which I have already sent to my students, so they have the access to the PowerPoint. However, I am going to record the lessons by explaining on each slide. For example, in this topic, which is related to transcription and gene expression, the PowerPoint slide already prepared, the essential idea and the information that they need to know is prepared for each slide, and also you can see at this slide that they need to know what is expectation, understanding, applications, and skills that they need to learn by the end of this unit. And then I start to explain slide by slide. And the reason is I am giving this introduction at the beginning of this video is because the video that you are going to watch after this is a very summarized part of one of my lessons. So you may feel that something is missing from the past or in the middle or something for the future. So because I just picked up small parts of one of my lessons so that I don't need to make it a very long recording, and you just need to know how does it work and how I prepare a lesson. Thank you for watching. Hello everyone. In this video we are continuing the discussion on transcription, but with a little focus on the nucleosome structure. And then we continue with the DNA methylation and acetylation. But before we move on to the topic of methylation and acetylation, I am sure that you have seen this slide before. Let's have a quick review on the topics over here. Okay, from here, the conversation and discussion on behalf of methylation and acetylation starts. What does that mean? Methylation is the addition of methyl groups to DNA. So you can see if this is a DNA and then the methyl group has been added in different parts of the DNA. So first step, what is methylation? Methylation is the addition of a methyl group to the DNA. What is acetylation? Acetylation is the addition of acetyl groups to histone. So here we go. This is the difference. In acetylation, acetyl group will be added to histone protein. As you can see over here, histone has different types of proteins, and this is the acetyl group which has been added to this H1 histone. Before I move to that slide, can you read this question and think about and pause the video? What evidence can you find that supports the idea that the environment can trigger heritable changes? Investigation or research which is called Lick Your Rats. You need to pause the video and read the text by yourself. Okay, did you do it? So, I'm not going to read everything here. The experiment is that there are some types of rats that they are licking their pups, and some of them don't. And apparently, for those rats that they are licking their pups get some epigenetic inheritance from the behavior of their mother, and that's so interesting and very lovely, findings from this research. Those rats that got licked very well by their mom, they are so calm. But those rats that they are not licking their child or their pups, then pups getting more anxious, and they show more anxious behavior while they're growing. This it depends on the environment and depends on the situation. It's not that if it's the anxious rat is a good or bad. It depends on the environment. We are going to talk about the environment effect on the gene expression. Look at these two cute rabbits. So cute. What do you think the difference between these two? So you see that they have different colors around the nose, and also ears are a little bit different. These two have the black color, but this is no. What do you think? Do you think that they have the genetic differences? Pause the video and a little think about that. Okay, tell me what was the result of your thinking. Let's see. The environment of an organism impacts gene expression. For example, human hair and skin color are impacted by the exposure to sunlight and high temperatures. Okay, so what does that mean? This means that they have the same genetic information, but some environmental factors have changed that. Explain how the temperature will affect the expression of gene. What gene? Gene C. So what does it mean? There is a gene which is regulating the presence of the skin color in the form of black color or white color. If the gene which is controlling the fur pigmentation in Himalayan rabbits, both of them are Himalayan rabbit, this gene, when the temperature is higher than 15, and it is between like 15 to 25 degrees centigrade, gene C, you tell me, gene C, what happens to gene C? Gene C is inactive. It's not active. So when the gene C is not active or is inactive, it doesn't make the color, it doesn't make the skin color to present as a black color, which means that it doesn't activate the pigmentation. So that's why there's no pigmentation, there's no black color happening here. But in the other side, or in the other hand, low temperatures, which happens at this rabbit, the gene C becomes active. So kind of gene C is in love with cold temperature. Even you can remember or memorize something like gene C loves cold. So gene C, who loves cold, means that in the colder temperature, it is becoming active and it starts to create the pigmentation. So that's why you can see these cute area of the nose and ears become a black color.